Hello everyone, and welcome to Clinical Research Statistics for Non-Statisticians, presented by Roe. I'm Michael Gibney, editor for Fierce Life Sciences, and I'll be moderating this webinar. Our speaker today is Jennifer Marcello, Senior Biostatistician for Roe. You can read her full bio on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the Download Slides widget at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentations by a Q&A session. Please submit your questions during or after the presentations using the Q&A box at the right of your window. Okay, now let's begin. Jennifer, please go ahead.
Hi everyone. Today we're going to discuss a variety of topics related to the role of the statistician in clinical trials, including clinical trial study flow, trial planning, sample size and power, hypothesis testing and p-values, data capture, randomization, statistical analysis plans, interim analyses, database lock, and then final analysis and reporting. The first step is planning your trial. This process starts as you begin your clinical program and anticipate what trials you may need and evolves as results from early trials come in. Once you've decided on starting a clinical trial, then you will write the protocol, develop a plan for randomizing subjects to treatment, develop and review your case report form and data collection methods, develop a detailed plan for statistical analysis in your statistical analysis plan, and design and create analysis data sets, design and create statistical displays like tables, listings, and figures. These are sometimes referred to as TLFs or TLGs. And finally, consolidate and report all procedures and results in the Clinical Study Report, or CSR. And here is your first pop quiz. During what phase should a statistician become involved in the process of a clinical trial? SAP writing, protocol design, CRF design, or study planning? You can choose on your screen which response you think is best, so I'll give you a few seconds to respond. Okay, it looks like the majority is correct. You want to involve your statistician at study planning. While it may seem like you really don't need a statistician, so you need a sample size calculation and statistical section for your protocol, as I hope you'll see as we proceed through this presentation, is that statistical considerations for a clinical trial have a major effect on trial design and endpoints chosen, so a statistician should be involved as early as possible. Now that we're starting to plan for our trial, we need to ask ourselves some key questions. What is our goal? What data do we need to collect to support this goal? And once we've collected this data, how do we test them to show whether or not we've achieved our goal? As an example, let's assume we're planning for a phase 3 randomized controlled trial to support a new drug application. And first, we need to describe the indication we are hoping to treat with our new drug. In this case, we're going to treat osteoarthritis knee pain with our drug, which is a new analgesic. And now we need to define the goal of our trial. And our goal is to show that our drug works better than placebo. To show this, we need to collect data on each subject's pain level. And using a visual analog scale, or a VAS, after 50-foot walk test is a common outcome for pain in osteoarthritis. Visual analog scales are used to have subjects rate their own pain on a scale from no pain to pain as bad as it could possibly be on a pre-measured 100-millimeter line. The subject marks on the line where they rate their pain, and the length between the start of the scale to the mark is measured in millimeters to estimate their level of pain. For this study, we're going to ask our study participants to rate their pain after a walk test on multiple study visits throughout the duration of the study. Given that plan, let's go through some of the questions that the study statistician may have to further define the design and analysis of our osteoarthritis study. First, we need to define the population as specifically as possible. For our study, we want to enroll subjects who have osteoarthritis of the knee. But we also want to ask ourselves if we're looking for subjects with arthritis that is unilateral in one knee or bilateral in both knees. We also may want to ask ourselves if we want to include all ages or if we want to focus on a particular age group of arthritis patients. Next, in order to evaluate the appropriate type of statistical analysis methods, we need to determine what type of data we are collecting. In this case, we are estimating pain from 0 to 100 millimeters on a visual analog scale, and so this is considered continuous data. Continuous data is any data that can be measured on a continuum. Other examples of continuous data are age, height, weight, blood pressure. Any outcome that can take of any value within a range is considered continuous data. We will discuss other types of data further later in this presentation when we talk about statistical analysis plans. Now that we have a type of data, we also need to know how many time points we have. As we mentioned earlier, we are planning to rate the subject's pain level at multiple study visits throughout the study. So we will need to do what we refer to in statistics as a repeated measures analysis. So now that we know what our outcome and our number of time points, we need to determine what is the best test to use. Given this information, we have a continuous endpoint with repeated measures, so we decide that we will use the difference of the least squares means between the active treatment and placebo group, and we will use a repeated measures population average model to estimate the least squares means difference. Next, we should decide if we want to plan on performing any sensitivity analyses. We may decide that we want to perform analyses on pain reduction in each knee separately and compare it to pain reduction in both knees. We could also perform sensitivity analyses evaluating different methods for dealing with missing data. And the next consideration we need to make when planning a trial is whether or not we will be blinding or masking the treatment assignment in any way. There are three main types of study blinding. Single blinding is when only the study subject doesn't know which treatment they have been assigned to, but the investigator does know. This may make sense when the investigator giving the treatment needs to know what the treatment is, for example, in a trial testing different surgical procedures, but we still want to reduce the subject's bias of perceived benefit. Double blinding is the most common type of blinding in randomized controlled trials. In this case, both the subject and the investigator do not know what treatment the subject has been assigned to. Triple blinding is when the subject and investigator do not know what treatment the subject has been assigned to, and the data monitoring committee also is kept from knowing what treatments the subject has been assigned to. The primary reason that we mask or blind participants and investigators is to reduce bias. Although at the start of the trial we really don't know whether or not the new treatment being tested is better than control, whether that control is a placebo or another treatment, it is human nature to assume that the new treatment is better, and knowing the treatment assignment could influence the subject's perceived benefit as well as the investigator's perception, of whether the subject is being helped or not. This bias can influence the response to treatment in ways that may not accurately reflect reality. The disadvantage to blinding is that some subjects may not want to consent to be involved in the trial if they think they may end up on placebo or what they perceive as a worse treatment. Also, keeping the appropriate people blinded to the treatment assignments can be complex, and appropriate procedures need to be put in place so the blind is not broken. For triple blinding, not knowing the treatment group could affect how well the data monitoring committee can assess patient safety. In order to keep the treatment groups blinded, they may only review pooled safety and data, and thus wouldn't notice if there was a trend towards a particular adverse event in one of the treatment groups. In some cases, blinding is not really possible. For instance, if you're comparing a daily tablet with a biweekly intravenous treatment, then it would be difficult to blind the subjects to which treatment they're receiving without subjecting them to a lot of unnecessary dummy treatments. And also, just to note here, although we're talking about blinding subjects and investigators, in reality, most of the clinical study team should remain blinded to treatment assignments when possible to avoid bias in making decisions while the trial is ongoing and to reduce the risk of accidentally unblinding someone who should remain blinded. Now for our next pop quiz. True or false? I can keep only the subjects who improve on the study treatment in my analysis. This is, of course, false. You need to keep all the information you collect during your trial in order to accurately assess the outcome. So since you're keeping everyone, you want to give careful consideration to who should be in your study population, and this is done by defining inclusion and exclusion criteria to select subjects that are representative of the population you are interested in treating with your new therapy. Referring back to our osteoarthritis example, we would not want to include subjects who had osteoarthritis of the hip but not of the knee, since we are specifically interested in osteoarthritis pain of the knee. We might also want to give careful consideration to whether we want to include subjects who had osteoarthritis of both the hip and the knee, if we want to focus on subjects with only osteoarthritis of the knee. Once we define our study population, we need to appropriately define our analysis sets. Common analysis sets are also called analysis population. They include the safety analysis set, the full analysis set or ITT population, the per protocol population, and for some indications, a clinically evaluable analysis set may be defined. The safety analysis set is anyone who received at least one dose of study treatment. This set is used for all safety analysis reporting on outcomes like adverse events, clinically significant vital signs, and laboratory test results. Our full analysis set is also referred to as the intent to treat or ITT population. It is anyone who is randomized to study treatment, whether or not they actually received treatment with either the study drug or the control. They should be included in all analyses of drug efficacy. A per protocol analysis is generally done as a sensitivity analysis to see how the efficacy results look when only subjects who completed the study as planned are included. Per protocol analysis sets should be defined on a study level and may exclude subjects who are not compliant with the study drug, who dropped out early, or who had major protocol violations, such as being randomized despite not meeting all inclusion or exclusion criteria. Now that we know who we want, we need to figure out how many subjects we need. If we do not enroll enough subjects, then we may not be able to adequately address our study goals. However, if we enroll too many subjects, then we can waste time, money, and other resources. In addition, we may be exposing patients to a drug that doesn't work, so we want to be considerate of that and not enroll more subjects than we need for our research. All types of clinical trials should include some justification of sample size. However, it's not always necessary to do a formal power calculation. Power calculations are used when you are planning to use a statistical hypothesis testing and may not be applicable for some phase 1 and phase 2 trials whose goals may be to assess other outcomes such as safety, pharmacokinetics, or maximum tolerated dose. As an example, we may use a precision estimate for a phase 2 trial where the primary goal is to estimate safety. In this case, our sample size justification would be something like, with a sample size of 40 subjects, the probability of observing a toxicity that occurs in at least 5% of the time is 87%. However, for a phase 3 randomized control trial, we almost always need to define a primary hypothesis and choose a sample size based on adequate power to assess that hypothesis. Power is the probability of seeing your treatment work in a given study, assuming that it does work. Power and sample size calculations are dependent on the planned analysis method and the hypothesis you're testing, so this needs to be decided before sample size calculations can be performed. You also need to determine what is the clinically significant difference that is needed to show that your treatment is better than the control. Statisticians sometimes refer to this as an effect size, and it is important for the statistician and clinical team to work together to decide on effect size that will provide clinical benefit to patients in the target population and is achievable given your previous clinical and non-clinical study results. Also important are type 1 error, or alpha, which is the probability of a false positive result. That is the probability of saying the treatment works when it doesn't. And type 2 error, or beta, which is the probability of a false negative result. That is saying the treatment does not work when it does. We also need an estimate of variability for our primary endpoint, and for this we use the standard deviation to estimate the variability of a response variable. Other important considerations when deciding on a sample size include cost, as more subjects will increase the cost of the trial, and the expected dropout rate. It is good practice to inflate the sample size you need in order to make sure that you have enough subjects, even if there are dropouts, to ensure that at the end you have enough power to detect a difference in your treatment groups if there is one. Sample size and power have a direct relationship. The higher power desired, the larger your sample size will need to be, and the smaller desired power, the fewer subjects you will need in your trial. That is, a sample size with 80% power will be less than a sample size for a study that has 90% power. Your sample size will also increase with more variable responses, variables, as indicated by larger standard deviations. Your power will increase with smaller acceptable error, that is your type 1 error, and smaller desired clinically significant differences. In contrast, your sample size will decrease with less variable data, that is smaller standard deviations, larger clinically significant differences, that is it takes fewer subjects to show a difference between treatments of 20% than it takes to show a difference of 5%, and with larger type 1 error rates. In power calculations, you will specify the desired power and acceptable error. And for phase 3 trials, it is common to estimate sample size using a power of 80 to 90% and a type 1 error rate of 5%. The clinically significant difference in standard deviation will be dependent on your indication and the endpoint you are using to measure response. And also, of course, cost will increase as the sample size increases, so you need to keep that in mind as you make estimates for your sample size calculation. We've been discussing statistical power and sample size relative to hypothesis testing, and now I want to spend a little bit of time talking about what a statistical hypothesis is. Earlier, we used an example of a trial to compare a new treatment to placebo to treat pain due to osteoarthritis of the knee. When we define a statistical hypothesis, we are turning the primary goal of our study into a null and alternative hypothesis. The null hypothesis is always that there is no difference. So in our osteoarthritis example, the null hypothesis is that the least square mean is the same in the group that received drug as in the group that received placebo. Our alternative hypothesis is that there is a difference in the least square means for the placebo and treatment groups. This 2x2 table shows how our hypothesis relates to type 1 and type 2 error. As we mentioned previously, our type 1 error is our false positive rate. It is the probability that we reject the null hypothesis given that the null hypothesis is true. Remember, if our null hypothesis is true, there is no difference in the treatments. And our type 2 error rate is the false negative rate. It is the probability that we do not reject the null hypothesis, even though our null hypothesis is false. That there really is a treatment difference, but we do not reject the null hypothesis of no difference. Recall that power is the probability that says our drug works and it actually does work. This is 1 minus the type 2 error rate. It falls into the bottom right quadrant of this 2x2 table and represents correctly rejecting the null hypothesis when the truth is that the treatment works. It may be useful to use the analogy of a smoke alarm to help you remember this. Your alpha or type 1 error is a false alarm. Your smoke alarm goes off, but there is no fire. Your type 2 error rate is a broken alarm. There is a fire, but the smoke alarm never goes off. So remember, the lower the allowable error or type 1 error rate, the larger sample size you'll need. The more statistical power your trial has, the larger sample size you'll need. The more variance or larger standard deviation will also result in a larger sample size. And the larger your desired clinically significant difference, the smaller the sample size. Now for another pop quiz. Another true or false. A statistically significant difference is all I need for the FDA to approve my treatment. Oh, good, 100% correct. So this is, of course, false. You need to show not only that there are statistically significant results, but also the result is clinically significant. Remember that sample size is related to the size of the clinically significant difference that you choose, so it is possible to show that even small differences are statistically significant if your sample size is large enough. It is important that you choose a clinically significant difference that will also translate to benefit for patients. Next, we're going to talk a little bit about p-values and statistical significance. Last week, the American Statistical Association issued a statement regarding the use of p-values, and this is the official definition of a p-value from that statement. It is, informally, a p-value is the probability under a specified statistical model that a statistical summary of the data, for example, the sample mean difference between two compared groups, would be equal to or more extreme than its observed value. That is, the p-value is the probability of observing a value as extreme or more extreme than what was actually observed if the null hypothesis is true. Recall that alpha is our type 1 error rate. If our p-value is greater than alpha, then we cannot reject the null hypothesis, and the result is considered statistically insignificant. And if a p-value is less than or equal to alpha, then we can reject the null hypothesis, and our result is considered statistically significant. We set the alpha to be small enough so that we can be reasonably confident that when we reject the null hypothesis, we are doing so because our treatment actually works. The more times you test your hypothesis, for instance, by doing interim analyses, the greater the chance you will reject the null hypothesis when there is, in fact, no treatment effect. This is also true for testing more than one hypothesis by having multiple primary endpoints. There are statistical methods for taking this into account, but they will affect your sample size calculations. So if you do need to perform an unblinded interim analysis, or you have multiple primary endpoints, it is important to plan for this before starting your trial. Okay, so once you've finalized your protocol and determined the appropriate sample size, you're ready to start planning for data collection. The CRF, or case report form, is where all the data are collected for the trial is defined. The statistician, in addition to other members of the clinical team, should review the CRF. The statistician is going to be particularly looking to make sure that all the data necessary for planned safety and efficacy analyses are collected and that they are in the appropriate format. Once a trial is underway, it is very difficult to go back and get the data you need, so carefully planning and review of the data collection procedures are key to a successful trial. If your trial will be part of a submission, you also need to think about if there are any data standards that you may be required to follow for particular regulatory agencies. It goes without saying that during clinical trials, we want to avoid missing data whenever possible. Good protocol design and data monitoring procedures can help avoid major missing data issues. However, sometimes you will still have missing data due to subjects missing visits or not completing all of their assessments at a visit. If you anticipate a large amount of missing data for your study endpoints, you should have a plan in place for how to deal with this during data analysis. There are many methods available, including missing data imputation methods and sensitivity analyses. Industry-wide clinical data standards are becoming more widely used in the clinical trial industry, and the FDA has a strong preference for submissions that use these standards. This is a broad topic that we don't have time to discuss in detail today, but I just wanted to mention a few common data standards briefly because you're likely to hear these acronyms as you work in clinical trials. CDISC stands for the Clinical Data Interchange Standards Consortium. It is a nonprofit organization that is dedicated to establishing standards for use in clinical research. CDISC standards include C-, which are standards that are used for data collection and CRF design, SDTM, which are standards for organizing the raw clinical data that comes out of the database into data sets that will then be used to create analysis-ready data sets. ADAM standards provide guidelines for creating data sets that are organized to easily perform the statistical analyses and data summaries needed for your data analysis. The CDISC website on the bottom of this slide, and you can check it out if you are interested in learning more about data standards in clinical trials. For clinical trials involving more than one treatment group, randomization is an important tool to reduce study bias. Since allowing the subject or investigator to choose the treatment may result in biased allocation, such as preferentially giving the new treatment to subjects that may be perceived to have a greater benefit, it is important that this process be done in a random fashion. Randomization also has the benefit of producing treatment groups that are roughly equal in the distribution of baseline prognostic factors and demographic characteristics such as age and race. The first type of randomization we're going to discuss is simple randomization. It is the easiest type of randomization, and it is like flipping a coin. However, the risk is that you could randomize everyone to the same treatment group. After all, even though it is rare, it is possible to flip a coin 100 times and get all heads. To avoid this risk, we can use a permutated block randomization, where the treatment groups are assigned in a random order for each of several blocks. In this example, we have a block size of 3 and 3 different treatments. In each block, the order of treatments is randomly assigned. This method is very good at producing balance across arms. You can see here at the end of each block there will be the same number of subjects in each of the 3 treatment groups, A, B, and C. However, if you know the block size, you could guess the next treatment. For instance, if you know you are randomizing the 9th subject tomorrow in the above design with a block size of 3 and the subject you randomized yesterday was in group C and the subject randomized today was in group A, then you can deduce that the next subject will be randomized to group B. You can also use stratification factors at randomization. is useful if you want to make sure that you randomize enough subjects within a particular subgroup, such as disease severity. In this example, a stratified permutated block design is used to enroll subjects both with moderate pain and severe pain. The protocol has a section regarding statistical analysis, but it is typically fairly short on detail, especially for analyses other than the primary analysis. The Statistical Analysis Plan, or SAP, is where details of the planned analysis of both the safety and efficacy of the trial is recorded. Specific details on analysis methods, how missing data will be handled, and any other details that are necessary to carry out the statistical analysis at the end of the trial are included in your SAP. Here are some of the types of statistical tests that we use for different types of outcomes. As mentioned earlier, continuous data is any data that can be measured on a continuum, like age and height. We can summarize this data using descriptive statistics such as mean, standard deviation, median, and range. Some common hypothesis tests used for continuous data are t-tests and Wilcoxon tests, analysis of variance and analysis of covariance, that's ANOVA and ANCOVA, linear regression models, and linear mixed models. Categorical outcomes are data that can be placed in discrete categories. Demographic values like sex and race are categorical outcomes, and treatment response is sometimes evaluated using categorical outcomes. For instance, we may define subjects as either responders or non-responders to a particular treatment and put them into two distinct groups for analysis. Descriptive statistics for categorical data are frequency counts and percentages. Some common methods for hypothesis tests of categorical outcomes are chi-square and Fisher's exact test, the Cochran-Mantel-Haenszel or CMH test, McNemar's or Kappa's test of agreement, logistic regression models, and Poisson models. Survival analysis, also referred to as time-to-event analysis, is how we summarize and analyze data such as time to death or time to progression of disease. They are very commonly used in oncology trials, but can be also used to analyze any type of time-to-event data. Survival analysis is unique from other continuous outcomes in that censoring is taken into account. Censoring is when you know the subject has not met the event of interest at a particular point in time, but you do not know what happens after this time. At that time, the data is considered censored. As an example, you may only follow subjects for disease progression up to a certain amount of follow-up time after the last study treatment. Since you can't be sure that they did not have a disease progression after they leave the study and are no longer followed, the subject is considered censored at their last study visit if they have not had disease progression up to that point. Kaplan-Meier methods are used to compute descriptive statistics such as median survival time that take censoring into account. Because censored subjects can bias the results if not accounted for properly, it is not appropriate to use standard descriptive statistics for time-to-event data. You can use log-rank tests, survival rates, Poisson models, and Cox proportional hazard models to perform hypothesis tests of differences in survival between treatment groups. Sensitivity analyses frequently include comparing results when missing data is excluded from the analysis and to results imputing missing data using methods such as last observation carried forward, LOCF, baseline observation carried forward, BOCF, and multiple imputation. Pattern mixture models can be used to model response while accounting for differences that may exist between subjects who drop out of the study early and those that complete the study. Sensitivity analyses can also be performed on different populations of subjects, such as the per-protocol population, to see how results differ from the ITT population. Next, we're going to talk a little bit about different types of interim analyses. Your next pop quiz. I can look at my data whenever I feel the need to see if my treatment is working. Is this true or false? Of course, this is false. As mentioned earlier, the more times you look at your data, the more likely you are to incorrectly reject your null hypothesis. If you're going to do an interim analysis, you need to ask a few questions. The first is whether or not the interim analysis is planned. Next, what is the purpose of the interim analysis? At what point of the trial and how many times are you going to run your interim analysis? Do you need to unblind the data, and if so, is an unblinded team needed to run the analyses and review the results? What is the state of the data at the time of the interim analysis, and do additional data cleaning procedures need to be put in place? And finally, how does this affect your type 1 error rate, and are any adjustments needed for type 1 error control? One of the most common types of interim analyses are data summaries prepared for data monitoring committees. The purpose of these committees is to review subject safety while the trial is ongoing. The frequency of these meetings is dependent on the duration of the trial and the anticipated safety concerns. You may need an unblinded study team to run the analyses and present the results to the committee. However, in some cases, the DMC remain blinded as well, so an unblinded team is unnecessary. You could arrange for interim database locks or just take data snapshots with the understanding that the results presented may have some data that has not been fully reviewed at the time of analysis. Generally, your type 1 error is not affected because these committees are looking primarily at safety data and the primary hypothesis test for efficacy is not performed. And sometimes you don't have a lot of information about your primary endpoint as you collect data, you find that the variation in the data is different than the estimate you used in your power calculation. In this case, you may want to perform an interim sample size calculation. It is not necessary to unblind the team, and alpha is not affected if you use a pooled standard deviation. That is, you calculate the standard deviation combining all treatment groups and assume equal variation for all groups in your sample size calculation. For some trials, particularly those running for a long time, you may want to perform an interim analysis to apply stopping rules. These are rules that allow you to stop the trial early if a predetermined threshold for efficacy or futility are reached. The timing is based on the primary outcome, and it is required to have an unblinded study team perform the analysis and review the results. You want your data to be as clean as possible and should plan for an interim database lock. This type of analysis will affect your type 1 error rate, and this should be accounted for in planning your trial. Adaptive design, such as pruning algorithms, which can be used to test multiple doses of study drug in the same trial, removing or pruning those doses that don't show efficacy as the trial progresses. Again, the timing is based on the primary outcome, and if the study is blinded, you probably will need an unblinded team to perform these analyses. You, again, want the data as clean as possible since efficacy decisions are being made, so you should plan for interim database locks. Your type 1 error will be affected, but adaptive designs are generally only used for phase 1 and phase 2 trials, whose goals are more to gather evidence to plan for your future studies and not to make decisions on drug efficacy, so it is not as much of a concern. Once we have finished data collection, before we proceed to the final analysis, we need to lock the database and unblind the data so we know which subjects received which treatment. For our final pop quiz, when is it appropriate to unmask or unblind a database for analysis? A, just prior to locking the data. B, just after database lock. C, whenever I need to see if my treatment is working. D, when all analyses are set. E, both A and C, and F, or F, both B and D. The majority of you got this correct. It is both B and D. We do need to perform all data cleaning and lock the database first, but we also need to have made all final decisions on all planned analyses to ensure that we are not biasing our results by picking analyses that will give us the results we want to see. So before we lock our data, we need to plan for all of our analyses. Also, if we are planning to perform any analyses on a per protocol population, the protocol violations need to be reviewed, and the subjects meeting the requirements for the per protocol population need to be pre-specified before we know what treatment they received to, again, avoid biasing our results. The statistician will usually sign off on the lock and confirm that we are ready to unblind. Before locking, we want to make sure that the data is of the highest quality possible and that all data should have been carefully reviewed and planned data cleaning procedures performed. All site queries should be resolved. If there is missing data, every effort to populate the data should be made before locking the data. And then finally, we will unmask the data so we can perform the final analysis. The last step is to perform all of our analyses as specified in the statistical analysis plan. Missing data are imputed if necessary. Secondary response outcomes and safety data are also summarized. And finally, we can perform our hypothesis tests on our primary endpoint or endpoints, and we find out if our drug worked. The last step for finalizing a trial is to write a CSR, clinical study report. The protocol and SAP become part of this document, and all the results are described. Study procedures are detailed, and any deviation from the planned study are described along with how they were managed. All of our tables, listings, and figures are included, and explanations of key results and what they mean for our drug development are included in the report. And then that's it. Our trial is finished. So, in summary, it's important to give careful consideration to determine the trial objectives early on. The type and design of the trial should be aligned with the overall clinical plan for the drug's development. Determining the sample size is an important factor in the cost of the trial and requires deciding on your statistical hypotheses during the planning phase. Statistical considerations for a clinical trial drive many trial activities, including sample size calculation and data collection and data cleaning procedures. And in order to reduce bias, all analyses should be pre-specified. This slide references some documents and books that you might find useful if you want to learn more about statistics and clinical research and how good statistical practice helps ensure successful clinical trials. And now we can take some questions.
Great, thank you, Jennifer. Now let's move on to the Q&A. Reminder to please submit your questions during or after the presentations using the Q&A box at the right of your window. We have lots of great questions already, and we'll try to get to as many as possible. Let's go here. Okay. What is the pro and con in changing primary endpoint in an ongoing blinded late phase study? How often does this happen?
It shouldn't happen. It happens very rarely. Because, as mentioned, all your sample size calculations and your planning and your data collection are all dependent on this primary endpoint. So you might have to go back and redo quite a bit of work if you change your endpoint, especially if you're changing it to an endpoint that you didn't already collect. So the risk is that you change your endpoint and you find out at the end that you don't have the appropriate power with the sample size you have, and that if you don't have appropriate power, you may actually have a treatment effect, but you won't be able to show that statistically, and then your trial fails and you've wasted a lot of money. So the only, definitely consult with your statistician about whether, if you feel like this is important to do this. And of course, and actually at this point, you should probably be talking to the FDA as well.
Great, thank you, Jennifer. All right, next question. Major protocol violations are those which could affect analyses of efficacy and safety. What are examples of major and which are examples of minor? Are there gray areas? Should statisticians be consulted to correctly identify those in the gray area?
I think the statistician and the clinical team needs to work together to determine which are major and minor. You know, if somebody is randomized but they didn't meet all the inclusion and exclusion criteria, they should be included in your analyses, but it might depend on how much this is really going to affect your outcome. A lot of inclusion and exclusion criteria are based to protect patients because the drug is unknown. For example, it's pretty common we don't want to include any pregnant patients. So, you know, a pregnancy might be a protocol violation, but that may not affect the outcome of your efficacy results. Minor violations would be something like missing a visit, which happens, not completing, you know, having missing data, not following the drug compliance could be major or minor depending on how far off a patient's compliance is. If they miss a couple of days, that's probably not a big deal, but if they're consistently not taking the drugs, then that's probably a major protocol violation. Generally, when there are protocol violations, the entire clinical team, including the statistician, will discuss them and determine whether they should be considered major or minor.
Great, thanks. Next question. If an interim analysis is performed but no changes are made, why would this increase the type 1 error rate for the final analysis?
Because every time you look at the data, you know, basically when you do an interim analysis, you're looking at the data. You don't have all the full information available, and then you're going to look again. And this kind of it's sort of your second look is not independent of the first look, and that just basically it's a very mathematical phenomenon, but it increases the probability that you will see a result. You'll reject your null hypothesis when you shouldn't. So it's really based on looking at the data, not whether or not changes are made.
Oh, I'm sorry. I had my
oh, sorry.
button on. I apologize. If p-value of the primary endpoint is not significant, but the secondary endpoints are statistically significant, and there is a clinical benefit, can the drug still be approved?
If you pre-plan it that way, possibly you could try to convince the FDA. There's certainly procedures where what we do is you test the primary endpoint first, and then if it's not significant, you might test the secondary ones. But usually your secondary endpoints are chosen such that they're kind of dependent on the primary endpoint or they're related. So if your secondary endpoint, it really depends. Most likely not. You might have to, you should choose your primary endpoint because that is the response you're looking for. For many indications, the FDA has preferences on what their endpoint response variable is considered standard in the industry, and so you may or may not have that much choice. If you do have choice, for instance, you're studying a disease that not much is known about, then you might be, what you might want to do is go ahead and just do another trial using your secondary endpoints, or at that point you really should talk to the FDA and see what they think.
Okay, thanks, Jennifer. How might using LOCF versus VOCF change the results of the safety analysis?
It may or may not. Baseline observation carried forward is sometimes considered a little bit more conservative than last observation carried forward. But it would be highly dependent on the outcome and how much imputation you're doing. But it actually, usually you're not going to impute data for a safety analysis. It's more used for efficacy analysis.
Great, thank you. Next question, how might the ITT analysis versus per protocol analysis change the results of the study?
Well, you probably need to show that it works for the ITT analysis, and if you could come into an instance where the ITT population doesn't show a significant effect, but the per protocol population does show an effect, you've probably got a design or administration of drug issue that you might need to go back and modify, do another trial where you modify how patients are treated. Maybe if, for instance, your per protocol population is including people who took the drug, and they're supposed to take it twice a day, and you included everyone who took it twice a day, but you have a significant subset that kept forgetting, then maybe you would want to change your formulation so it could be a once daily drug or something like that. But ideally, you want the ITT analysis to match the per protocol analysis. That's what you're hoping for when you do these sensitivity analyses. Because we all are human, and people don't always take the drug as prescribed, and that's why the ITT analysis population is important, because it seems to it would reflect reality better than a per protocol analysis.
Great, thank you. This next question begins with a compliment. Excellent overview. In your experience, how involved is the trial statistician in the actual authoring of the methods and results section of the clinical study report? Thoughts on optimal level of partnership with medical writing team?
Usually, if you have, good medical writers can take your statistical results and write them very well. So the stat becomes an appendix in your CSR. So that part is authored by the statistician. But usually what the statistician's role would be to review what the medical writers have written and give comments in the draft. And definitely at that stage, working together to answer the medical writers any questions that come up as they're looking through the tables and listings and such. And so the actual text is usually written by the medical writer, but it is carefully reviewed by the trial statistician.
Great, thanks. This next one, please explain what is modified intent to treat analysis. Modified
Modified intent to treat. Sometimes you might, because of your outcome, you might change the definition of the intent to treat a little bit. I'm trying to think of an example. Like say, so you have your intent to treat analysis, but your outcome is, say it's an oncology study, your outcome's tumor response. Even if somebody got randomized, if you never took their baseline tumor measurement, you really can't do your outcome. You can't measure response on treatment. So the modified intent to treat might be all subjects who are randomized that have a baseline tumor measurement. So it's usually very much related to kind of clarifying the endpoint to make sure that you have at least enough to calculate it.
Okay, thanks, Jennifer. Next question. I am always intrigued by how the baseline characteristics across treatment groups always match so well with randomization. Is that always the case?
It's nearly always the case. Is that just a side effect of randomization if you're randomly choosing people? Generally, for large trials that are international, you do some stratification by clinical site or region, which really takes care of a lot of the variability that you might see by country or by hospital. But yeah, it's sort of a side effect of randomization. If you randomly choose people, then when you get everyone balanced out, you end up with pretty equal age and gender and race based on the underlying population. It should be pretty close to what those outcomes are in your underlying population.
Great, thank you. Next question, why don't we see statistics, p-value, etc., reported on safety data, and I apologize, Jennifer, I'm not sure how to pronounce this, N-Haste 3?
Oh, in phase 3, okay.
Oh, in phase 3, okay.
Yeah.
Okay, thank you.
No problem. A lot of it is because of that multiple testing. You know, if you were to run analyses on all your different adverse events, some of them are going to pop up statistically significant just by chance, and you're trying to avoid that multiple testing artifact. So that's the main reason, and it's just generally not. It's just generally not required for safety analysis. It's enough to just report counts and percentages. That'll give you a sense of the trend, and you can see any safety signals just by reviewing the descriptive statistics.
Great, thank you. We have a couple more questions. When is a crossover trial the best choice for trial design? Could it be used to compare the efficacy and safety of two routes of administration, using each subject as his or her own control?
Yes, you can. That would be one good use of that. I think it makes crossover trials. They have a lot of benefits. People act as their own control. The risk is that you need to make sure you have a washout period in between, so you aren't seeing the effects of the second. In the second period, you aren't seeing the effects of whatever treatment was given in the first period. And for some different types of drugs, that might not be really useful. Again, for some indications, it may not be ethical to do a crossover design. You might really need to treat everyone from the beginning. Like you generally don't give people who have like cancer or HIV placebos. So it could be useful. That the risk is that, and then the other risk is that people don't finish the second period, or they don't go into it, and then you don't have the full data.
Great, thanks, Jennifer. This will be our final question. We had a lot of great questions today and couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. What does the hazard ratio for a progression-free survival curve mean clinically?
The hazard ratio, so if you had a hazard ratio of, say, 2 comparing treatment, so the hazard ratio is 2 comparing treatment A over treatment B, it would mean that the risk of progression is 2 times greater for treatment A than treatment B. So it is a comparison of the entire progression-free survival rate in each treatment group. It's not linked to a specific point in time.
Okay, thank you, Jennifer. And thank you all for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speaker for participating and Ro for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining, and we look forward to seeing you at future events. Hello, everyone, and welcome to Clinical Research Statistics for Non-Statisticians, presented by Roe. I'm Michael Gibney, editor for Fierce Life Sciences, and I'll be moderating this webinar. Our speaker today is Jennifer Marcello, Senior Biostatistician for Roe. You can read her full bio on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to